Awesome. I would like to invite our next speaker up. So we are starting a new section of the agenda around STXBP1 in the lab. And our first speaker is Anna Carreras Mascaro from the Netherlands. She is from Matthias Verhaege's lab, and I am hopeful that sometime in my life I'm able to pronounce his name correctly, but he has been one of the longest standing researchers on STXBP1, and we're really excited that Anna's going to talk about variant spectrum and function. A lot of people have questions around, about the classification of their variant that they got in their variant report, and whether missense variants are different from others. And so her lab is working on getting some answers to those questions. Thanks, Anna.
Yes. Yeah. Thank you, Charlene, for the introduction, and the organizers for inviting me here. It's really exciting. So my name is Anna Carreras Mascaro, and I work in the Center for Neurogenomics and Cognitive Research in Amsterdam. I'm a neurobiologist, and today I hope to give you an update on STXBP1 variants and their function. So as you all know by now, STXBP1 is caused by variants in the STXBP1 gene, and what this gene does is generating the protein that we call MUNC18. Variants in the STXBP1 gene are known to cause a decrease in MUNC18 levels, and this is what we call in the lab and in the clinic haploinsufficiency. But what is MUNC18? Why is it so essential? MUNC18 is necessary for neurons to communicate from one to the other. So if we imagine a neuron that is sending a signal and a neuron receiving this signal, the signal itself is packed in what we call synaptic vesicles. And these synaptic vesicles are transported from one neuron to another in a very specialized compartment in the neuron that is called a synapse. In this synapse, the signals are sent from one neuron to the other, which has the receptors for this signal. And if we zoom in even further, we can see where the synaptic vesicle is being released, and there's a group of proteins that we have studied a lot by now, and one of these proteins that is essential for the synaptic vesicle to be released is MUNC18. But what does it mean that neurons communicate with one another? How do we study this in the lab? So in the video that I'm about to play, each of these structures that you will see lighting up is a neuron. And in this video, you can see how they are active, they are lighting up. You can see how these neurons are lighting up at the same time. This is because they are connected, they are synchronized, they are communicating with each other. But what happens when we don't have MUNC18? So we take these neurons in the lab, we remove all MUNC18, and this is what we see. So you can see that these neurons are trying to make a signal, they are lighting up, but you can see that they are not lighting up at the same time, they are not connected. And this is what we mean when we say that MUNC18 is essential for neurons to communicate with each other. But now if we go back to STXBP1, when we look at people, we have the DNA packed into chromosomes. And the DNA is structured in a way that there's fragments that we call genes. One of these genes is STXBP1, and as I said, this gene is necessary to make the protein MUNC18. So in a healthy individual, we have two copies of the gene STXBP1, and both of these copies are necessary to make the protein MUNC18. What happens in an STXBP1 patient is that one of these genes, one of these copies, has a pathogenic STXBP1 variant, and this copy is not able to generate as much MUNC18 as is necessary for the person to be healthy. And how do we study this in the lab? So in this picture here, this is a picture from a microscope, and here we have neurons in magenta, and you can see how they have these extensions, and that's how they connect with each other. In this type of experiments, we can also look at MUNC18. which I now included also in this picture. It's in green, so all the green that you see in this picture is MONK18, and the blue dots that I hope you can see are synapses. And these neurons come from a patient with STXBP1. Now, what happens when we look at also neurons, but from a healthy individual? If we look at MONK18, you can see how it's clearly brighter if we look at healthy neurons. And when we quantify this is how it looks like. In this graph, we have MONK18 levels in neurons, and you can see how STXBP1 patients have lower MONK18 levels than healthy neurons. And this is why we believe that the disease is caused by not having enough MONK18 in the neurons. So the neurons are not able to communicate like a healthy person would. Now, STXBP1 variants, the story is a bit more complicated. Not all the variants in STXBP1 are pathogenic. There are actually a lot of variants that we call benign. So these are differences in the gene, but this gene can still produce the right amount of MONK18 protein. Then we, of course, also have the pathogenic or likely pathogenic variants. We call them PLP. These are the ones I just explained that cause a reduction in the levels of MONK18 and cause the disease. But there's a third type, which are called variants of uncertain significance. These are variants that are found in the gene in a patient, but it's not clear whether they cause the disease or not. If we look at the STXBP1 gene, this is usually how we visualize it with the different pieces. And if we look at the variants that have been described today, along the gene, there's actually more than 1,000 variants that have been described so far, but many of them are benign, many of them are pathogenic, of course, but there's also a lot of them that we don't know what their effect is on the protein. We don't know if these variants will cause STXBP1-related disorder or not. So as I mentioned, we more or less understand what happens when a variant is pathogenic. We have a reduction in MONK18 levels. We also more or less know what happens with benign variants, but with variants of uncertain significance, it's a bit unexplored. We really don't know what they are doing and where these variants are coming from. This is usually a patient and their family that go to the clinic, they get genetic testing, they receive the news that they have a variant of unknown significance, or they might not even get this news. Maybe the clinician keeps this news for themselves because at the end of the day, they don't get a diagnosis. They go home without knowing what disease their child might have. And this is why we decided to start this project that I will show you today. And this is in collaboration with BioMarine, which is a sponsor also of today's summit. And BioMarine is a biotechnology company that focuses on rare diseases and is doing a lot of work for STXBP1. And together with our lab in Amsterdam, we want to have better evidence to say when a variant in STXBP1 is pathogenic or not. So to improve classification of variants and hopefully improve the diagnosis. So in this project, we looked at 400 STXBP1 variants and their effect on the protein, which is ultimately what will have an effect on the neurons. So with this project, we hope we can help resolve variants of unknown significance. We hope we can increase the number of patients with a proper genetic diagnosis. And we also want to help understand even further the disease. In this project, we started with 400 variants from different classes. So we started with 17 variants, 263 variants of unknown significance or conflicting, and 120 pathogenic or likely pathogenic variants, or PLP. And as you can see in this scheme here, this is a scheme of the whole MONK18 protein, and the variants are annotated along this sequence. You can see how the variants that we selected are distributed along the whole protein sequence. Most of the variants that we studied have never been looked at before. Some of them have, which also helped us understand the experiment better, and I will show you some of that in a bit. And how did we do this experiment? So we first introduced STXBP1 DNA, and from that DNA we then measured how much protein was being made. So for example, we always introduce also a healthy copy of the STXBP1 gene. This is what we call in the lab wild type, or WT, which you will see in the graph later. And then we measure how much protein this healthy gene is producing. And then we will do the same with when we introduce an STXBP1 copy with the variant. And we will compare how much MONC18 is being produced in the wild type situation and in the variant situation. And I will be showing you now the results in this very large graph that I will explain a little bit first. So at the bottom here, I annotated all of the variants, which it's not really easy to read because it's 400 variants annotated in this axis. And in this graph, I will be showing you MONC18 levels that we detected for each variant. And the wild type condition that will be shown in red is it's set at 1, so it's a bit easier to understand the comparison to wild type levels. And this is how the data looks like. As you can see, the benign variants are in green, and all of them are above this 0.5 line, going towards the wild type levels. But if we look at the PLP variants, the pathogenic variants in purple, they are clustering mostly below this 0.5 line. So there's a clear distinction between pathogenic variants and benign variants. And the VUS and conflicting variants are distributed along the whole intensity level, MONC18 level profile. I also annotated here with the red dot the recurrent variants. These are variants that are found in more than 10 STXBP1 patients. You can also see how they are more towards the lower levels of MONC18. But to look at those a bit more closely, I also put them in this graph. Now the graph is inverted, so it's a bit easier to read the variants. And if you can see, the wild type levels are at 1. You can see how all the recurrent variants have MONC18 levels below 0.4. Now, if we take this data and I show it in a different way, I can also separate the classes into different graphs, like you can see here. And what we can see from this graph is that all of the benign variants are, again, above 0.5 levels of MONC18. If you look at the PLP variants, they are clustering below 0.5. It's most of these variants, 82.5%. So there's a clear distinction between benign variants and PLP variants. And then if we look at VUS conflicting variants, we can see that there's a lot of variability with the levels. But there is this group here that the levels of MONC18 are very similar to the levels in the PLP. This is 99 variants that we can hopefully help reclassify with this assay. And we can hopefully, to some of these patients, give finally a diagnosis. So if I summarize all of the data that we have produced and that I have shown, we started with 17 variants, and all of them had either MONC18 levels very close to wild type or in the intermediate cluster, but we did not find any benign variant with low levels of MONC18. If we look at the VUS conflicting variants, they had more variable levels, of course, but what we can take home is that 99 of these variants have very low levels of MONC18, which could indicate that they are actually STXBP1 patients that never received a diagnosis. And finally, we can see that most of the PLP variants showed low levels of MONC18. So to summarize, most of the PLPs that we have identified presented very low levels of MONC18, which even strengthens even more our hypothesis that STXBP1 is caused by loss of MONC18, which we call haploinsufficiency. And this is also strengthened because we saw that the recurrent variants that are found in many patients also show reduced MONC18 levels. We also provide evidence to hopefully help resolve 99 variants of unknown significant or conflicting. And we also want to add even more variants to this type of work, and we also will select some of these variants and take them to neurons and study them more deeply. So basically what we want to do with work like this is take the data that we produce together with genetic data, structural data, and clinical data, and hopefully give a more confident and complete answer to the patients and their families. And with that, I wanted to start by thanking all of you, the STXBP1 community, patients and parents, and all the caregivers. My lab, of course. I also want to especially thank Lotte. She did all of the lab work that I showed today, which was a lot of work. And the biomarine team, we couldn't have done this without them. And I will be taking questions gladly.
Thank you, Anna. Thank you. This is really important work, and as Anna outlined, there are a number of patients who get there are a number of individuals who get a variant of uncertain significance on a genetic report, and that clinically is kind of you're not diagnosed with the condition, and so this can really help us to help those individuals understand where they are and potentially become part of this community. So really important work. Does anyone have any questions? We have time for 1 or 2 questions. So there's a question back here.
Hi, my name is Marlena. I have a son who clearly has STXBP1. I have a question. I don't know if you could answer this. I know you stated that you need a certain amount of protein to have this and then something else to have this. Is there any correlation between like having a very hard pregnancy, being on multiple medications, and them actually kind of like experimenting on you that can kind of alter things or, change things up a little bit? Is there any research on that?
Mm-hmm. That's a very important question. We are currently, we have a collaboration with a clinician that will help us disentangle all of these things, because of course, as people that work in the lab, we don't have access to patient information because it's confidential. Mm-hmm. So we will work directly with a clinician that will help us answer questions like this. I think it's extremely important.
Yes,
yeah.
He was my first child, and I was actually on bed rest like the whole 9 months, and I was on a lot of medications that I don't think I was supposed to be on, had a lot of things that were being done that shouldn't have been done. So I'm just interested to see if anything could have altered how he was developing.
Yeah, I was not aware of these issues also during pregnancy, but I'll make a note of this. I will also ask the clinician if they have this information.
I suffered from hyperemesis gravidarum, and my case was very extreme. I probably would throw up maybe like 30 times to 50 times a day. I lived on IV fluids. I had an in-home aid. It was a lot.
Yeah.
Yeah, thank you.
Yeah, thank you.
And I will ask someone from the STAR or ESCO team also. I'll come back to you on that, because in terms of the medical history that's captured. Thank you so much, Anna.
Thank you.
And